As Elvisha said, the kind of big question that if you wanted to summarize what we are doing in my lab with one big question, it's how does the brain work? And so, of course, looking at the hierarchy of the brain, the kinds of methods and models and things that you use to answer this question is going to depend on the level at which you're interrogating the brain. And of course, there are many levels in the brain. Today, what I'm going to be focusing on mostly is this kind of this lower right-hand corner of looking at whole brain in vivo neuroimaging in humans. And that could either be, analyzing voxel level data or analyzing groups of voxels like brain regions, or looking at interconnectivity between brain regions and how those change with disease or behavior. And then also looking at how the brain interacts with its environment. So the lower right-hand corner is generally what my lab focuses on. And that's what I'll be talking about today. Okay, so the talk outline, basically, I'm going to start with some discussion of general concepts in brain imaging, including connectivity, connectome measurements. So I just want to briefly review that because that's what the majority of the talk is going to center on. So I'll kind of briefly go over those concepts and then look at how machine learning can help us with brain behavior mapping or in understanding how the brain works in healthy control populations, including encoding and decoding models or neural control. Yeah, if I were going to be salacious, I would say like mind reading and mind control, but I will not be salacious. All right, so largely brain behavior mapping. It's essentially where you observe some cognitive or physical behavior from a person, testing their abilities cognitively or behaviorally in some other way. And then they also undergo an MRI exam at the same time. And then, of course, depending on the MRI protocols that you use, you can extract different types of biomarkers, including maybe tractography, looking at structural connectivity, or fMRI, resting state task-based, where you look at physiological connectivity networks in the brain. Or they could be things like, PET imaging or ASL perfusion or QSM. So there's lots of different modalities and ways to measure the brain. But essentially what you do is you extract these biomarkers from imaging and you do some sort of modeling, either statistical modeling or machine learning to map brain and behavior. So I see kind of two large applications of this. One is a clinical application where you're trying to either diagnose a disease, prognose how a person is going to recover after injury or disease, and maybe even use it in a treatment selection capacity. And so looking at the basic science part of that is looking at brain behavior relationships in healthy controls to identify the underpinnings, either anatomical or physiological, in some certain behavior that happens in a healthy control person. Okay. So I'll talk a little bit about the connectome because a lot of the work that I'm going to discuss is based around the connectome. So there's a lot of work and effort into mapping the connectome in the human brain. And when I say the connectome, I largely mean two different types of connectivity networks. One is anatomical, where we're looking more at diffusion MRI and tracing white matter fibers through a process called tractography. So looking at structural and anatomical white matter connectivity networks in the brain. And then the other is more physiological, where you're either measuring directly neuronal activity using EEG or MEG, or you're measuring a proxy bold imaging using fMRI. And there, what we're doing, instead of extracting the strength of anatomical connections between brain regions, here we're trying to identify co-activation patterns over time in different regions of the brain. Okay, I apologize if you hear some noise in the background. My daughter's doing something out there. Okay, so I'm not going to go too much into this, but essentially this is a slide showing us how the structural connectome is extracted from the brain using a process called diffusion MRI that tracks diffusion of water molecules in the brain. And of course, you can think of an example of two extreme cases where you have free water in a particular voxel MRI. And then you have a white matter tract that has some structure to it. And of course, the water molecule in the water molecules are going to diffuse equally in all directions when you have free water. And then they're going to diffuse along these structures. For example, in a voxel that's in white matter, has a white matter tract through it. Okay, so that's essentially how you can measure anatomical connections in diffusion MRI. And here I just have a video at the bottom showing you some scrolling through pairs of gray matter regions and showing you the white matter that connects those two gray matter regions. Okay. The functional connectome, as I said, it's looking at coactivation patterns over time, either using fMRI, EEG, or MEG, and they're looking at basically patterns of coactivation over time. They can either be task-free or task-based. And of course, there are some limitations. This also exists in diffusion MRI too, some physiological noise, motion and dropout, and of course, multiple corrections comparison. There's this interesting dead salmon experiment where a dead salmon was put into MRI and they showed some activation of voxels in the dead salmon. Which obviously was not happening, but the statistical underpinnings of it are very delicate. You have to be careful about those multiple comparisons because you are measuring from a lot of different voxels in the brain. So the last decade has basically seen an explosion of brain behavior mapping. So mapping connectome quality to abilities in healthy control individuals, looking at creativity, IQ. There's this really seminal work in the field looking at how learning juggling over a 6-week period could actually change the anatomical connections between different regions in the brain. So it's not just that your connectivity is mapping to function, your function can also feed back to your connectivity and change it as well. And then, of course, the same thing with functional connectivity, looking at how that relates to different cognitive abilities has just exploded in the last decade or so. On the flip side of that, looking at how disconnectivity can relate to impairment is also an important thrust of connectivity analysis, and looking at how disconnections due to strokes or lesions or injury can cause cognitive or motor disabilities. It's really interesting there. So the picture I have there is probably you guys know Phineas Gage. Of course, so there was this really cool study by Jack Van Horn where they took his skull and kind of recreated where the railroad spike would have gone, and then showed that the disruption to the structural connectome was actually very detrimental in that area in relative to the same size disruption in other areas. So it's kind of an interesting study there with the classically known case in neuropsychology. Okay, so getting to machine learning. So of course, anytime you have big data and large dimensionality, one option is to use machine learning. And people have started using a lot of machine learning for supervised classification or regression techniques where you want to diagnose certain neurological conditions or psychological conditions, or even looking at health variables across healthy control populations like age, sex, things like that. And of course, there's also the unsupervised approaches that you can implement in machine learning, looking at clustering. So maybe there are certain subclasses within a certain disease state that might be more similar to each other. I'll talk a little bit more about that in a second with depression. But trying to use machine learning to uncover, to either help clinical predictions or diagnosis or uncover underlying mechanisms within the population. And then there's this kind of interesting figure that I like to show that shows, publication year and classification accuracy. And as you can see, it's kind of decreasing, but a lot of that is due to the sample size. So the radius of these spheres are the sample size. And as you get larger samples, your classification accuracy can sometimes suffer because you have less overfitting. So some idiosyncrasies of connectomic data, especially when you're working with machine learning, of course, is this high dimensionality, small sample size, especially in clinical populations where you don't have a lot of subjects maybe that you're enrolling, but you're measuring a lot of variables. So of course, we have to be careful with those types of things, especially working with clinical data as class imbalance. So you might have some diagnosis that doesn't happen very frequently. So maybe you only have 7 out of 1,000 people with some diagnosis. And so you have to be careful statistically when you're doing that. Of course, there's special network topology, the connectivity networks, that isn't reflected in some machine learning strategies like CNNs and things like that. Some advantages of using connectomic data is that comparisons across subjects is trivial. I say that, but it's a little more nuanced, but I'll just say that because it's the easiest thing to say. But basically, my dorsolateral prefrontal cortex is similar enough to Bobby's DLFPC that we can, call it even. And each node actually has a biological meaning. So that's also helpful in interpretation. And then, of course, some disadvantages of connectomic data is there is heterogeneity across subjects and across time even. And sometimes these labels are relying on noisy clinical measures of diagnosis or prognosis that may not be super accurate. And of course, the model is only going to be as good as the data that it's trained on. Okay, so machine learning applied to clinical populations is especially important when you have some disease that's difficult to diagnose, but may benefit a lot from early intervention. So things like autism or developmental delays, things like diseases related to aging, like Alzheimer's, Parkinson's, and frontal temporal dementia. So early intervention is key there, and it's not is very easy to diagnose. And then, of course, depression is another thing. I'll talk a little bit more about that in a minute, but treatment of depression is quite difficult. So we could possibly use some machine learning techniques to identify these subpopulations that might benefit more from one treatment versus another. And then looking at how connectivity maps to these disease impairments might actually give us some insight into developing new treatments. So if we know that, strengthening one connection between two regions is going to cause a better outcome, then we might focus on developing treatments to actually improve that connection. So it might give us some ideas about future treatments, or even looking at genetic biomarkers that are underpinning some of these diseases. So there's this kind of a cool paper. It's 4 years old now, but they looked at reproducibility of biomarkers from fMRI, and here they were undergoing a classification task of trying to classify autism in typically developed individuals. And this is the ABI data set, which I don't know if you all know about it, but it's a large data set of autism fMRI and typically developed fMRI data. It is quite noisy, and it's collected from different sites. Those sites have different imaging protocols, and so it's kind of complicated to combine. But there are some architectures and ways that you can deal with different sites, so data from different sites using machine learning. So here they were just basically showing that if you performed intrasite and intrasite and intersite cross-validation, you kind of got different results. But it was an interesting study nonetheless. And then, of course, how you process your data and extract your biomarkers can greatly impact the accuracy of the classification as well. So looking at atlasing using different gray matter structures, gray matter atlases, the number of regions obviously has an impact on the classification accuracy. How you calculate correlation of the connectivity matrix, and then of course how you model the mapping between biomarkers and behavior, they can all have impacts on that. So that's another thing to be aware of when you're working with machine learning applied to neuroimaging data. Then there's this really interesting paper from our Connor Liston's group where they were looking at disease subtyping in, so they had fMRI in almost 1,200 subjects, which is really a great undertaking. And they performed unsupervised clustering, and they actually revealed 4 subtypes of depression. And each of these subtypes, they tended to have different imaging and clinical phenotypes in each group, particularly in the limbic and frontal striatal areas. And so they extracted them from one site, and then they actually used that cluster information to classify out of sample subjects. And then they found a really high sensitivity and specificity for that classification. And they also used this approach to identify those depression subjects who might respond to TMS. So it was actually, this is an example of how you can use machine learning to identify subjects that could benefit from a certain type of treatment. And then there was this really interesting study looking at the Connectome fingerprint in antidepressant response. And so they were actually saying that you could use baseline measures of functional connectivity to identify people who would respond to drug interventions. So it's kind of interesting there. We also had a project where we looked at classifying autisms versus typically developed individuals. And there we used kind of this idea of stochastic parcellation, which instead of relying on some predefined atlas, we did like kind of a randomization of combining different voxels that were nearby each other into certain regions to get an atlas, and showed that when you use this type of approach, you can actually get better classification accuracy than you can when you rely on a single atlas. So it's kind of interesting there. Then we also looked at how you can use baseline demographics and imaging biomarkers to predict therapeutic gains in chronic stroke. So this was a chronic stroke subjects that were enrolled and underwent TMS and robotic therapy. And we were actually able to predict with pretty high accuracy how well they would do after the therapy. So this is also another example of how you could use this to identify people who are going to respond better to certain treatments. And this is another work, sorry, my previous slide was also from Jaron Toslu, postdoc in our lab. And this is also work that we've been doing with a collaborator of mine, Susan Gauthier, where we're looking at classifying disability versus no disability in multiple sclerosis patients. And there we were actually able to classify pretty well if a person had a disability or not. And then looking at the importance of the variables in the model, we isolated the left cerebellum as being particularly important for having structural connectivity in the left cerebellum to be very important in predicting whether or not a person had disability. And the disability was, much of it is driven by motor disability, so it kind of makes sense in the context of the cerebellum. So with that, I will say that I really enjoy applying machine learning to clinical populations, but the sample sizes are quite small generally in clinical data. And of course, this is a nice figure showing that it's nice or depressing, depending on your viewpoint, looking at the sample size versus out of sample replication, so accuracy. So you can see that even when you have a few thousand subjects, many times you might not even be able to predict so well the out of sample population. So that's I'm motivating this talk by saying that looking at control populations is a little bit easier because you do have some big data sets that are available in control populations, particularly things like the HCP, Human Connectome Project, where there's a ton of connectivity imaging data available for these subjects and demographics and other types of behavioral cognition scores. And so one of the things that we do is apply machine learning to these control populations, these huge data sets, to identify possible demographic differences like sex differences or age differences, and do brain behavior mapping with cognition or other behaviors. Some other types of applications are, classifying into resting state versus task using the fMRI data, predicting functional connectivity from structural connectivity. And then, of course, there's encoding and decoding models. The encoding models our goal is to predict the brain's response to a particular stimulus. One example I can give you is images. So when a person views an image, you could use an encoding model to predict how their brain is going to activate. And then, of course, the other opposite direction is decoding. So using the brain activation pattern to predict the stimuli. So that's cool too. And then looking at fine-tuning stimuli to get a particular neuronal response that you want. So I'll talk a little bit about that. So this is obviously one of our hosts, Elvisha. Her most recent work, it's under review right now, but we were using functional connectivity and structural connectivity to predict cognitive scores. And there we used, obviously we used the machine learning approach. And we showed largely that functional connectivity was more predictive of cognition, which was kind of interesting. And we were able to isolate, in particular, functional connectivity in the subcortical structures of our atlas that we used. And structural connectivity between cerebellum and limbic structures seemed to be important also in predicting cognition. So this was kind of a really interesting work that's currently under review. Again, with Elvisha, we looked at time series analysis of fMRI data in healthy controls, and we actually showed that there are sex differences in the temporal dynamics of functional MRI signals. The thing that we looked at was Hurst Exponent, which is kind of a measure of temporal memory. And there we were able to use this measure of temporal memory to classify males versus females in our HCP data. So it's kind of revealing some possible sex differences in temporal dynamics of fMRI data. A really recent paper that just came out is looking at predicting functional connectivity from structural connectivity using a neural network. And there they showed, I'm going to move this because this was an animation, so I'll just move that one over. So here we have on the lower left-hand corner, the empirical functional connectivity. So anytime you see EFC, it's the empirical observed functional connectivity, and the upper right-hand corner is the structural connectivity. And you can see with the biophysical model that they used, that's the second one over, it was a neural mass model. They didn't do as well in predicting functional connectivity as they did when they used the neural network. So this is actually showing that using machine learning, specifically neural networks in this case, had a higher accuracy at predicting functional connectivity from structural connectivity rather than a biophysical model, neural mass model. And there they were actually able to show that so the loss function they used was maximizing the correlation between empirical and predicted functional connectivity, but also trying to match the predicted functional connectivity for that subject versus every other subject. So kind of trying to enforce some sort of individual identifiability, and that I think was their contribution to the literature on that. And they were actually able to show that, unsurprisingly, their predicted functional connectivity did a better job of predicting cognitive outcomes than the predicted functional connectivity from the neural mass model. It didn't do quite as well as the empirical functional connectivity, but it did better than the biophysical neural mass model. So it's kind of an interesting project. Another project that I've been working on with Meenakshi Kholsa and Mert Sebanchu and Keith Jamieson in my lab is this brain encoding model. So here we had the HCP movie watching data. And what we did was we used different types of neural networks to extract data from the visual images that are presented, the features from the visual images, and then also the features from the auditory stimuli. And we used either 1 second or 20 seconds of data in this prediction encoding model. And we were actually able to show pretty good agreement with the actual brain activity for the visual and the auditory and the multimodal models. So we're actually able to do a pretty good job of predicting how the brain responds to the movie, the visual stimuli from the movie and the auditory stimuli from the movie. And interestingly, we showed that, you know, the auditory regions did a better job of, we did a better job of predicting the activity in the auditory regions when we used the auditory input versus the visual input, et cetera. So it was a really, really interesting project with that group of people. Okay, so getting to brain decoding. This was kind of old work right now, but I really liked it. It's 4 years old, but they did 129 minutes of natural video with 7 subjects. And they had WordNet that actually labeled every 1 second of data with the content. So they had a word that described the content of that image. Then they built a hierarchical logistic regression model for every category, and they used 5,000 voxels out of the previous 4 time points and 5,000 bold voxels. They used the bold images from the previous 4 time points to predict the category of what the person was looking at. And so the results, this was kind of interesting. So it was accurate for some things more than others. So for example, this top panel here is for the category talk. And so for the category talk, we have the blue is the predicted probability that the model predicted that content was talk. And then the gray is when it actually happened. And you can see obviously that they do a pretty good job. The AUC was 0.91. For things like that are kind of more amorphous, like thing. So of course that's a little harder to predict rather than something that's very typical. And we know that there are certain brain areas that activate when a person talks in a video. But it's kind of an interesting example of how you can use brain activation patterns to decode the stimuli, properties of the stimuli. Taking it a step further, instead of just predicting the category, you can also predict the actual image that a person is looking at. So here is an example. This is a really recent work where they showed images and trained trained a neural network to reconstruct the image that the person was seeing from the fMRI signals. And you can see there's some agreement. It's not exactly right, but it is pretty close, surprisingly. And here I have a couple more examples. You can see, so on the left is the actual image that was seen. And then from the left to the right is, I think, increasing probability of that solution being the one that is true. So you can see, of course, some things, they look pretty close. There's some, the color is matching up, the shape of the object and the location of the object is sort of matching up, but it's not exactly the same, obviously. It'd be pretty strange if you were able to perfectly reconstruct it, but yeah, the work is going in that direction for decoding signals. Another very important application for a lot of people is decoding EEG signals for brain-machine interfaces. So there are people who are quadriplegic or tetraplegic, that no longer have control of their bodies, but there are brain-machine interfaces that people are using more and more frequently that will record brain activity from the person and control a robotic arm or control a wheelchair, or even in this case, let me see if I have the video, turn the sound off, is controlling an exoskeleton. So this is actually really cool. This person is a tetraplegic, but they underwent this training process to have a machine learning algorithm basically that records the electrical activity under these sensors and then puts it through a classification algorithm to detect what the person is actually wanting and what their desires are to walk or to move. And so, yeah, so this is so cool. So we can actually help a person that is not able to move, we can actually help them move their body. So I think it's pretty inspiring what people are doing these days with brain-machine interfaces. But a lot of it does rely on machine learning algorithms and interpreting the physiological signals from the brain into the physical realm. So let me just move on to the next slide. Right, so this is another area of research that I'm also interested in and maybe doing some experiments next year in, but it's neuronal control. So here, this is a really cool project. They were macaque monkeys and they had images that they were showing the macaque monkeys. And they had direct neuronal recordings of spike frequency of specific neurons, so single neurons. And they showed these monkeys these images, and their goal was to create an image that would maximally fire a specific neuron. So it would cause a maximal spike frequency for a particular neuron. And they used these, they borrowed this approach from machine learning, adversarial learning. So creating these synthetic images that can trick deep learning into classifying, say, like an image of a cat as a dog. So there's these interesting algorithms that can create synthetic examples to get a particular response that you want. And so on the right there is actually an example of the iterations over the training. Of course, as you present an image and you record the spike rate, then you can tweak that image and see if you get a better spike rate. And you have this iterative optimization, basically, procedure where you start from some kind of noisy looking image. And then you end up with, I think, something that looks pretty monkey face-like, right? So in the lower right-hand corner, you can kind of see this sort of monkey face emerging from that particular neuron. And so some data from that. The generation on the x-axis is like the iteration in this optimization procedure, and then the response is the spike rate per second. And you can see that the reference is in green, and then the synthetic, it starts out by the reference, and then it starts to increase, up to like 60 spikes per second, which is quite an increase from about 10 spikes per second. And they were able to show that, like the early synthetic images didn't do as well as the late synthetic images. And then they also had some natural images of human faces and monkey faces and things, but nothing ever got close to this response rate that they were able to elicit with the synthetic image. And then they they selected different neurons, and they did the same thing for different neurons within different monkeys. So here, this RI10 and RI12, I believe that's in the same monkey RI, but it's just a different neuron. And then there's Y1 and GU1, which is another monkey, but they were able to replicate across monkeys and also replicate within neurons, two different neurons within the same monkey. And it's interesting because the synthetic images that create this maximal spike frequency in one neuron from the same monkey is not the same as another neuron. So it's not surprising, but it's kind of interesting nonetheless. So the similar thing has been done. Again, they did it, I believe this was in macaques. And here what they were trying to do, they had two different cost functions. One of them was to actually just maximally fire a population of neurons. So in this case, it's not a single neuron that they're recording, but a population of them. And one of the objectives was to maximally fire a certain population of neurons. And another one was this kind of one-hot population control where they wanted to specifically maximize a set of population of neurons and minimize the activity of others. So a selective activation versus just a maximal activation. And here's a couple of examples from one of their populations. So the top row is the maximal activation that they could get, and then the bottom row is kind of this one-hot population approach where they wanted to maximize the frequency of firing in one set of population and minimize it in others. So you can kind of see the differences in those two objective functions. But it was a similar approach where they were synthesizing images to maximally fire a population of neurons. Okay, so I guess I went too fast on my other ones, but this is another one that's interesting. It's one of my last slides, but this is really recent. I think this just came out like maybe last week, but we can go through this a little bit. So essentially they had humans listening to a podcast, and they would stop the podcast. If any podcast fans are out there, it was This American Life. So it's kind of close to my heart. In any case, they would stop the podcast and ask the person if they could predict what the next word was in the podcast. And so there are language models in deep learning that can generate language, and they do a pretty decent job of predicting the next word in a sentence. And they actually do pretty similarly to humans. So that's what that scatter plot was. They had this language, hierarchical deep learning, deep language network, they call it, so DLN. And it did pretty well at predicting the next word in a sentence. And then what they did was they recorded using, I think this was ECOG in certain patients. So they had ECOG, I think, in epilepsy patients, because obviously you're not going to do ECOG in healthy controls. But they did ECOG in epilepsy patients, and they recorded their neuronal activity during this podcast. And they trained an encoding model that would take the neuronal activity and predict the words. Okay. And then they, the encoding model is predicting the neuronal, sorry, I got that wrong. The neuronal signals from the word embeddings using the different lags. So using like the neuronal recordings that, time further and further away from the onset of the word. And then they did a decoding model where they predicted the word embeddings from the neuronal signals. And they're actually able to show, interestingly, that this model trained on the ECOG data was able to predict even before the word onset what the word was going to be. So up to like 200 or 300 milliseconds before the word onset, it was able to predict accurately the word that was coming up from the EEG signal itself. So that's an example of how neural networks, machine learning neural networks, could be sort of insightful into actual neural networks. So there's this kind of intertwining of the two. Okay, so some conclusions are that machine learning applied to this neuroimaging data, which is going to become more prolific and more voluminous in the future. They actually can help us understand how the brain works, give us insights into how this very complicated organ works. And then, of course, in clinical applications, it can help with diagnosing these diseases that may benefit from early interventions. It can help us prognose outcomes after things like traumatic brain injury or multiple sclerosis. It can also help us actually perform therapy for people. So in the case of the brain-machine interfaces, of course, it can also help us select the treatments that are going to be more accurate for a particular person, so personalized treatment selection. And then also looking at treatment development, especially in the case of the depression, was pretty interesting. And of course, so a lot of this hinges on sharing data. So I'm always an advocate for large public data sets, or even sharing data through things like Enigma. So really helpful as well. I'm a part of that. And then again, I'll leave with a warning, is that your machine learning model is only going to be as good as the data you provide it. So if there's bias in your data, there's going to be bias in your algorithm. There's been a lot of talk and a lot of really good highlighting of how there may be bias that exists. And actually the speaker this morning was talking about that in this depixelation algorithm. So there was this algorithm that took pixelated faces and it was showing that it could like reconstruct the pixelation. But when you fed it faces of, say, a person like Barack Obama, who has dark skin tone, when you did an anti-pixelation of that, it ended up looking like a white person. So this is just an example of how your model is only going to be as good as the bias that exists in your data. So that's always something to be aware of when we're working with machine learning, especially in clinical populations where decisions that are made based on these algorithm choices might influence clinical care. Okay, so With that, I'd like to thank all of my, obviously, lab members, my awesome collaborators, and then I will do a shameless plug for Machine Learning in Medicine series that I was just talking about. So if you guys are interested in machine learning in medicine, and I will say that the two co-directors, myself and Mert Sebanchu, are neuroimaging people. So a lot of our series centers around neuroimaging. It's not exclusive to, but I would say probably like 80% to 80 to 90% are neuroimaging related. So you can go on our website and actually find all of our talks. They're recorded there. And you can email either Elvisha or I to get you added to the listserv. Okay, so that's all.
Yeah, thank you, Amy. I'm glad you also touched on how sharing large data sets is very beneficial.
Yes.
Cameron spoke this morning about his 1000 Connectome project and how he faced a lot of pushback at first about sharing data.
You know what, it's really tough, I think, especially for clinical populations. I mean, there are a lot of protections in place for clinical data, which is good because obviously we don't want to violate any ethical considerations. But as long as those considerations are met and approved, then I think there should be more push for sharing of these clinical data sets, because there are, you know, the number of subjects is obviously lower than healthy control populations in almost all of these diseases, right? So the only way that we can get to a point where you could use machine learning on some of these biomarkers is if you have these huge data sets. And I think Enigma is really helping in that. So there's different Enigma projects for different clinical diseases. But there still remains a lot of diseases that don't have large public data sets that are very heterogeneous and actually are kind of common. So like, I'll give an example of multiple sclerosis. So as far as I know, I don't know of any publicly available databases in MS. But this is a very, you know, it's a pretty common disease, and there should be a lot more publicly available data sets that will allow us to have insight to these diseases, because that disease is actually very heterogeneous and complicated and not easy to treat. So I think it would benefit a lot from some of these approaches.
Yeah, I think, is Dave still here? Dr. Kennedy is here. I think he's on Enigma too, if he wants to talk about the Enigma project.
I shared my stroke data with Enigma, the stroke Enigma. So we're doing some, actually, Emily, we had just applied to get access to all of the Enigma data for the stroke project. Yeah.
Oh, nice.
Yep.
Yeah. So, I don't know if Alicia has questions. I have one quick question, which is more, I guess, relevant to my curiosity. It's pretty obvious that like the dimensionality of these machine learning algorithms is hugely beneficial to decoding and encoding. Oh, Dave says, I don't know any MS public data sets. Sad face.
Yeah, I was in discussion with somebody from Australia who was also of a similar mindset. So we might be trying to get something together soon. So yeah.
So,
although it's, like I said, it's hard to get people to agree to share their data when they're collected clinically, and it does take a lot of blood, sweat, and tears to collect data in a clinical setting. So I'm not gonna shame anybody into sharing their data, but
so going back to what I was saying, so I guess, but in comparison to those biophysical models like the neuromass models, they obviously have interpretability benefits, but I'm just wondering, like, is there still efforts in pushing those physiological models for decoding, encoding, and like basically describing how the brain is working? And like, why would anyone want to use those when you can have this huge neural network trained on a GPU to do like really well already?
Yeah, I mean, I think I would say it's been a huge shift to using neural networks to do these encoding and decoding problems, because they just work so much better, like you said, right? But there may be some people who ultimately don't like the black boxiness of a neural network, even though they're not so black boxy, because there's actually been some evidence that when you're looking at specifically at encoding models, that some of the layers in the encoding model are actually reflective of the hierarchies in the visual processing system of the brain. So there's some, you know, some information that can be gleaned from the structure of these encoding models in the ML sense for the actual brain sense. Yeah.
Yeah. Gael is the author for that paper. He's in the Brainhack Mattermost if you guys want to ping him.
Yeah, it's
paper.
such a cool thing. Yeah, yeah. We're working with a collaborator of mine who's doing something similar to with CNNs and things. So we're looking at some cool images from that. So, or frightening, I guess you could say. They look a little frightening. They're kind of like those synthetic monkey images that are sort of haunting, right? Like they're some sort of swirled, distorted monkey face. Yeah.
Yeah. Okay. Those generative networks are always so trippy.
Exactly. Yeah. I mean, so they're getting better and better by the day, right? So some of these generative networks are actually creating some of the things that I saw that were synthetically generated. You could not tell that they were synthetically generated. They looked like natural images, especially the most recent kind of generation of these generative networks. Yeah.
It seems we have a question from Yared in the chat, and he asked, for the brain decoding study, where they can approximate the image a person is looking at from physiological data, have they tried it on patients with diseases such as autism to see what an image looks like for them?
That's such a good idea, actually. I don't know of any work that has done that. I think most of the encoding and decoding has been in control populations, just because I think there's a proof of concept that people want to know that it's, actually doing something correctly. But I think that's a really good idea, because you could maybe get some insight into the experience of a person who has some atypical development in visual processing. So that would be really interesting, actually. Yeah.
I had a similar question based on like the neural control images that you'd shown with the mice. So we know that like for machine learning models applied to, or like using computer vision, you can add like a tiny bit of noise to an image and all of a sudden it won't be able to tell whether it's like a cat or like a truck or something. But when it's a human, or I assume most animals looking at it, we're not processing that noise in the same way. So in the work that you're doing, like how do you go about identifying which aspects of the synthetic images to actually change?
That's a good point. I'm hoping that the encoding models that we're creating that are based on actual neurological signals don't have that same problem. Okay. I'm hoping, but I don't know for sure. But yeah, that's definitely something to consider. But I am hoping that I mean, it would be interesting to actually see if you could use the encoding model that you train on like neurological data, if you could like do the same thing that they do with GANs, right? Where you like sort of manipulate the image and see if you get a drastic difference in the predicted brain activation. That would be a really interesting project, actually. Do you want to work on that? Just kidding. You have enough on your plate. But I think that's a really cool idea, actually. My hypothesis would be that it wouldn't be as easy. Because I'm guessing that the noise in the mapping is not as delicate as it would be when you're looking at something like a model predicting categories and stuff like that. Yeah.
Right, yeah. And I'm guessing like things like the spatial positioning of the image and like things like that also won't have the same influence. And
right.
like even the quality of the image, like we're able to process, I'd say like much lower quality image than a computer vision model might.
Yeah, that's true. Yeah, I wonder, I feel like you could actually just test that with an encoding model that already exists by trying to like get a drastic shift in the neural activity from a small change in the pixels, right? That'd be really cool to see. Yeah.
If anyone else has any questions, you can put them in the chat or the Q&A box.
I'm gonna stop recording for now.